As far as the funding, you can see many different funding partners. Most of the money is in place. We're just waiting for one application from Ontario to be submitted later this year. Genome Canada is putting in the bulk of the funding at $5 million. Genome Alberta, as well as ALMA in collaboration, putting in approximately $2 million, and Genome Prairie about $500,000. The industry themselves, we see, put the Canadian Swine Health Board over $1 million, PigGen over $1 million, and then the USDA through their PRRS Host Genome Consortium putting in close to $2 million, and the same consortium on circovirus is putting in just over $500,000 as well. The bioinformatics and some of the analysis for the gilt acclimatization project was put in by the Rosalind Institute in Scotland, and very happy to have that renowned institution being part of this project as well. This is almost too small to read, but the take-home message in this particular slide I wanted to show, along the left-hand side, there are 4 discrete projects, and we'll go into those in a little bit more detail. And essentially what those 4 projects are doing is generating tissue samples and data that can be put into a very large database and put those tissue samples into a number of different laboratories across Western Canada to do ongoing or further phenotyping or genotyping. And all of that will be managed through this big database, which in fact is run both in the US and at the University of Alberta. So the first 2 projects, very briefly, are on PRRS and circovirus, but they use the feeder pig. And in pig terms, the feeder pig is about a 20-kilogram pig after it has been raised through the nursery stage. So they'll be susceptible to disease, and both of these models are essentially inoculating a susceptible pig at about 20 kilograms and monitoring that pig over 5 weeks of age until it is terminated, and looking at various outcome measures, including levels of virus in the blood of the pig and in the tissues of the pig, as well as some production data. And in this case, for the PRRS, one of the more important ones they're looking at is average daily gain. And in circovirus, one of the ones, the early ones they've looked at are antibody production. So there's many different outcomes in both of these projects. And I'm going to show you a bit of data here that is very interesting. These are both ongoing projects, so they have some preliminary data coming in from the first set of pigs, and then we're expanding these projects to include more pigs, really so we can improve the genomics aspect of this. This chart here, and you see the traditional 4 quadrants, what we are looking at is levels of virus found in the blood along the y-axis, and they go from low to high. And along the x-axis, we're looking at daily weight gain. And what we see, if you look at the top right-hand quadrant, those are very favorable conditions, because those are pigs that grow fast, so high levels of average daily gain, in spite of having high levels of virus in their blood. And that, to me, is an animal that's potentially resistant to PRRS infection. It can perform very well, even though it's got high levels of virus in its blood. The opposite is true as well, and those are the pigs in the bottom left-hand quadrant, which actually have very low levels of virus in their blood, but they're also growing very poorly. And I think this whole project then is really geared up to look at the genomics of why those animals exist in the top right-hand corner. High levels of virus, yet high average daily gain. And this is a chart, and there's a frequency diagram, and what we're looking at is many different histograms there from the top down to the bottom, and that's different days that average daily gain or body weight was actually measured in these pigs from the day of inoculation through to day 35 or 40, which is the day of termination. And you can see the spread in body weights or average daily gain there, from a fairly tight looking histogram at the top to a very, very wide looking histogram at the bottom. And what we really wanted to look at here, or what these series of histograms really show, are the black bars, which are the littermate control. So those are animals that are not infected with PRRS virus. And you can see most of those animals are on the right-hand side of our histograms, meaning that their body weight or average daily gain is higher, which you'd expect because, in fact, they're not inoculated with PRRS virus, and they're littermate SIBs. But what's important here is some of the animals which are inoculated, represented by gray, are growing at about the same rate as the non-inoculated animals. So again, it's just more information to say there are animals there that are resisting virus or PRRS virus infection that we need to capture. We've done some preliminary work on genomics, and I'm not a genetics person at all, but I'll do my best to explain what this represents. We're using the 60K SNP chip in this particular experiment, and what we're seeing on this is the frequency of some of these SNPs that are showing up in these pigs. The chart on the left is the SNPs associated with viral load or levels of virus in blood, and the chart on the right is representing the SNPs associated with weight gain. And I think what the geneticists are really finding interesting is, in fact, we've got this SSC4 gene or SNP, which is in fact showing up on both. And I think they're quite excited that in fact they may have found something that is correlating to both weight gain and viral load. So more data in the future will hopefully clarify what those associations actually are. Switching gears now, looking at the circovirus model, and again, this is the same type model where we're inoculating and monitoring those pigs after infection. This particular chart is showing the development of antibodies after animals are inoculated with circovirus. And there's one antibody that's typically produced very soon after infection. It's called IgM, and that's the antibody that we're looking at in this chart. And we typically see IgM about 5 to 7 days after infection. And what this chart is actually showing is there's a lot of variation, and that's demonstrated by the 3 different colors on the screen. So the blue are the early responding pigs, and they're responding between 7 and 14 days. So that's about the time when we would expect to see IgM, maybe a little bit later than normal. But PRRS is not, or circovirus is a bit odd in that. But we also have this group of pigs represented by green, and those pigs are responding very late. So something potentially genetically different with those animals. And then what's very, very interesting is we've got these groups, this group of animals which is represented by red, and they're not responding at all by producing no IgM whatsoever. So 3 different categories of pigs there responding differently to the same disease model. So the question is, what is genomically different amongst these animals? And we haven't got that far, but hopefully as time goes on, we'll be able to pick up something that's significant. Okay, the third project is the gilt acclimatization project, and this one is led by the industry, i.e. PigGen, through their 9 members, representing the 95% of the Canadian breeding stock sold in the country. And what we aim to do is place about 4,000 F1 gilts. So this would be gilts that are 5 to 6 months of age, moving into commercial farms to be part of their breeding program. And in the industry, what we recognize is most of those gilts acclimate very well and become very productive females, but there's always that 5 to 10% that do not, and they either succumb after introduction, or I guess are poor performers or are called out before they meet their prime breeding parities, about 3 to 4 parities after introduction. So what we're aiming to do in this particular project is put 4,000 F1 gilts into commercial farms that are challenging from a health perspective. And in doing that, we're hoping to see some survive, but obviously some do not do well, they perform poorly. And then we'll compare the genomics of those animals that survive versus, and perform well versus those that perform poorly to see if we can pick out genomic differences. The fourth and last model, animal model that we'll be using is what we call the pregnant gilt model. And in this particular model, we are also incorporating a study of epigenetics. So we have a problem, or potential problem in the swine industry, in that our litter sizes are larger and larger. It's not uncommon now to have average total born litter sizes of 13 to 14 pigs born alive. And make a long story short, the uterus is not designed for that many pigs. So we have smaller pigs being born, birth weight is being reduced, and it's happening across entire litters. So the aim, one of the aims of this project is to, in fact, look at differences between F1 gilts that are born from high birth weight litters, comparing those to gilts that are born from low birth weight litters. So we've got that epigenetic component, which we're using the weight model, the phenotypic weight model to detect. And then we're using an inoculation model on top of that. So it's a 2x2 type experiment comparing high birth weight to low birth weight gilts, and also looking at susceptibility to PRRS virus when we inoculate a pregnant gilt at 80 to 85 days of gestation. Now, in that model by itself, inoculating pregnant gilts at 80 to 85 days is a complicated issue, because not only are you looking at outcomes in the dam, but you're also looking at outcomes in the litter. And in this case, there's 10 times as many feeder or little piglets as there are sows, so our number of sows and number of piglets becomes astronomical. Multiply that by the number of tissue samples that we have to collect, and we're into thousands and thousands of samples that we're hoping to collect and process. This particular model, then, we will use one particular breeding company, and just in the last couple of weeks, we're identifying the breeding companies that we'll use. They'll supply a pregnant gilt to us at 80 days of gestation. Term gestation in pigs is 115 days, so that's right at the beginning of the last trimester. We'll inoculate them. Hopefully the sows and the dams all survive, and then we'll terminate and collect the fetuses and sow tissues 3 weeks later, just prior to natural delivery, and send the tissues in multiple directions for multiple outcome measures. So we have a very exciting project just getting underway. It'll involve a lot of researchers. We're very grateful for the funding that ALMA has provided to us. We think it has some very distinct advantages and will benefit the Canadian industry. Synergistic, we've got tremendous ability to find powerful tools and put those into the industry. Given that we have an international consortium of renowned researchers and institutions, we think we have a tremendous likelihood of success. And more importantly, we anticipate we'll put tools back into the industry for adoption within 3 to 5 years. We estimate that we should be able to put over $25 million back into the pockets of swine producers to make a more profitable and competitive industry. Thank you very much. I appreciate the opportunity.